Hi, everyone, and welcome to the Reef Resilience webinar, Stony Coral Tissue Loss Disease, Lessons Learned and Resources from Florida. Thank you all so much for joining us today. I'm Liz Shaver. I'm the Science Lead for the Reef Resilience Network and your host for today's webinar. So all of our webinars and the rest of the Reef Resilience resources are brought to you through the support of NOAA's Coral Reef Conservation Program. And before we begin today, I'd like to go over just a couple housekeeping items. Today's webinar is going to be about an hour long. There will be a question and answer session at the end of the webinar, followed by an opportunity for additional Q&A online on the Reef Resilience Network forum. And I'll talk about that a little bit more at the end of this webinar. But in case you do have questions, there's two ways that you can ask them during this webinar and during the Q&A at the end. You can use the question box anytime throughout the webinar to send us questions, and we'll keep track of those for the end of the presentation. And you can also raise your hand. There's a little hand icon next to your name in your control panel for GoToWebinar. So you can click on this hand, and I will call on you and unmute you, and you can ask our expert presenters today your question directly. And if you're having any technical difficulties like trouble hearing or seeing the slides, please do send us a message via that same question box, and we'll try to work with you to resolve those issues. Okay, so this webinar on stony coral tissue loss disease is meant to be one of the first of a series because there are so many topics to cover and new resources coming out all the time. So of course, we can't cover everything in this webinar, but we would like to ask you at the end of this webinar what additional topics you would be interested in learning about so that we can better plan some follow-up webinars and make those resources really useful to you. So there's an exit survey at the very end. It's just 2 questions long. Please go ahead and take the time to answer those. Today's webinar has 3 presenters that we're really excited to have. They've all been working tirelessly to deal with this disease outbreak in Florida and have a lot of information to share with you today. So first, we're going to hear from Dr. Andy Bruckner, who is the research coordinator for NOAA's Florida Keys National Marine Sanctuary. He's going to share what's currently known about the disease from the latest science and research. Next, we'll hear from Maurizio Martinelli, who is the Florida Coral Disease Response Coordinator, part of Florida Sea Grant. And he's going to talk about Florida's really coordinated system for responding to the disease and how they work across agencies to share information and work together. And then lastly, we'll hear from Dana Wusinich-Mendez, who is the Atlantic and Caribbean Management Team Lead for NOAA's Coral Reef Conservation Program. She'll share information on resources that are available for the disease for people in the wider Caribbean in terms of detecting and responding to the disease. So I'm going to go ahead now and pass things over to our first presenter, who's Dr. Andy Bruckner.
Thank you for the introduction, Liz, and thanks to all of you for joining the webinar today. In 2014, a new coral disease that we've called stony coral tissue loss disease, or what I'll refer to as SCTLD, emerged within the Florida Reef Tract. SCTLD is the most devastating and longest-lasting coral disease that has ever been observed in Florida, and it's unprecedented in many ways. I'm going to describe very briefly what we have learned about SCTLD over the last 4 or 5 years, and also why it is so unusual. SCTLD, back 1 slide. SCTLD is the first coral disease that has shown a linear pattern of spread, with outbreaks radiating outward from its epicenter and progressively advancing both north and south throughout the entire reef system. It was first reported in September of 2014 off of Miami on reefs near Virginia Key, and within a year it had reached Palm Beach County in the north and Biscayne Bay in the south. Outbreaks occurred throughout Broward County and Palm Beach County in 2016, and it spread into Martin County, reaching the northernmost extent of the reef tract near St. Lucie Inlet by April of 2017. It also continued to advance towards the south, first appearing within the Florida Keys National Marine Sanctuary off of Careysport Reef in December of 2015, and during 2016 and 2017, outbreaks were reported throughout the Upper Keys. As it moved through the Florida Keys National Marine Sanctuary, the outer reefs were typically affected first, and then it would spread to the mid-shelf and inner patch reefs. By the fall of 2017, the disease was concentrated in the Middle Keys, and by April of 2018, it had advanced beyond the 7-mile bridge, emerging at Looe Key Reef. 9 months later, in January of this year, it first reached the reefs off of Key West. Another very unusual feature of this disease is the large number of susceptible species. To date, about half of the coral taxa that are found within Florida have succumbed to SCTLD. This includes the long-lived frame-building species, as well as 5 species that are listed on the ESA. Basically, this is the corals that make up most of the living cover on these reefs, with the exception of 3 groups: elkhorn coral, staghorn coral, and corals in the genus Porites. The third very unique feature of this disease is the way that it spreads among susceptible species. The disease typically first manifests on corals in the family Meandrinidae, or maize coral, followed by elliptical star coral and flower coral, and then it spreads to the brain corals. After several months, when most of the early susceptible species have died, the longer-lived frame-building star corals start to show the signs of this disease. Many of these star corals will exhibit new lesions that continue to occur on individual colonies over several months to years, or until the coral dies. When this disease first appeared off Miami, it was called white plague, which is another coral disease that had affected reefs in Florida and the wider Caribbean during the 1990s and early 2000s. While the initial disease signs may appear similar in some cases, and some of the same species are susceptible, the appearance of colonies is often dramatically different. If you look at the image in the lower right, this shows a coral that has signs of disease that are very, very similar to white plague, where the disease first initiated at the base of the coral, and it's progressively radiating in a linear band up across the colony surface. Unlike white plague, however, new centers of infection often erupt within the colony surface in areas that were previously completely alive. There's also a number of colonies that will show very unusual bleaching patterns that are adjacent to the lesions. The fifth feature that makes SCTLD unique is the high prevalence of corals that have succumbed to this disease within individual sites. For most other coral diseases that have been reported around the world, outbreaks generally affect between 5 to 20% of the corals of all susceptible species within one particular area. This figure illustrates the percent of corals that were observed with SCTLD within hotspots in the upper and middle keys during a single survey in either 2016 or 2017. On average, between 20 to 80% of the susceptible species will typically show signs of this disease at one time, and it continues to spread to additional corals within that reef environment over several months to years, ultimately affecting nearly every single colony of the susceptible species. To give you a sense of the spread within a reef over time, this is a map showing the spatial distribution of all the corals within a single plot. Each color represents a different species, and the white dots indicate actively infected corals, and the black dots are corals that died. So in January and February, the site was virtually disease-free, but beginning in March, the number of diseased corals rapidly increased, and by June, between 25 to 100% of the susceptible corals have become infected, and many of the corals have died. So diseased colonies tend to die within a few days to months, depending on their size. In this image, I've shown 2 photographs of a colony of Meandrina, or maize coral, that's about 60 centimeters in diameter. I photographed this coral in early April, and then again 2 weeks later. In this short time period, about 95% of this coral has died. Typically, diseased colonies have been measured to lose tissue at a rate of between 20 to 50 square centimeters per day, and the disease shows a linear advance across a colony of about 3 to 4 centimeters per day. We had hoped that many of the smaller corals of these susceptible species would resist this disease, but colony size does not seem to matter. Here are two different corals that both show active disease signs that are only about 3 centimeters in diameter. In every location within Florida that has been affected by SCTLD, the reefs have shown a rapid loss of live tissue area and a decline in the total number of corals within each site. This figure shows a series of graphs from permanent monitoring stations within Southeast Florida between 2013 and 2018. In the top graph, what you can see is the live tissue area for all the species within those permanent plots. The live tissue declined by about 40% from when the disease was first identified in 2014 to the surveys that were conducted in 2018. The bottom figures show the annual change in live tissue area, which are the colored bars, as well as the total number of living colonies within these permanent sites for 3 of the very highly susceptible species. In the 2 figures that are in the center and on the left, these are some of the early susceptible species, maize coral and elliptical star coral. These corals were virtually eliminated from these reefs within a year after the disease first emerged, while nearly half of the longer-lived star coral colonies, shown on the right, have died over this same time frame. A similar trend has been observed within the Florida Keys. This shows the percent decline in colony abundance between 2015 to 2018 within 40 permanent study areas in the lower, middle, and upper Keys. Overall, the highest proportion of losses were observed in the upper Keys. These are basically the gray bars, followed by the middle Keys, the red bars, and the lower Keys are the blue bars. The main difference that you see between regions reflects when the disease first emerged within that region. Basically, at the time of the surveys done in 2018, there were only a very small number of sites within the lower Keys that were affected by the disease, and hence there's been a much smaller decline in those susceptible corals. So I want to just switch gears and end my part of the talk with a little bit of information on what we've learned about potential causes of this disease. We know that the disease is caused by a living organism because it can be readily transmitted from a diseased coral to apparently healthy coral through direct contact. We also know that the pathogen is waterborne, and water currents serve as an important vector based on the way that this disease has spread throughout the entire reef system in the directions of prevailing water currents. There is also a considerable amount of evidence that a bacteria is involved somehow in this disease progression because we can effectively treat corals and eliminate the disease by using antibiotics. If we look at a cross-section through a single polyp of a coral that has a SCTLD lesion, what we see is that the disease typically starts near the base of the coral, adjacent to the skeleton, and it then progresses upwards towards the surface. In these early stages of disease, no microorganisms have been observed within tissue sections. In several species that have this disease, we've identified unusual crystal inclusion bodies that are found within the gastrodermis. These potentially may be viral in origin, but we don't know at this point in time. And we've also seen unusual bacteria within the coral mucocytes. If we look at colonies that have very rapidly advancing lesions, there are also a number of bacteria and other microorganisms that have been identified within the surface tissue layers. However, identifying these bacteria is no easy task. One approach that we've used to identify bacteria associated with diseased corals involves culturing both the coral tissue or the coral mucus within petri dishes that contain bacterial growth media. From cultures of 6 different species affected by SCTLD, 400 different bacteria have been isolated thus far, and these contain a number of pathogens. Some of these pathogens have been found in association with other coral diseases, as well as several pathogens that appear to be unique to SCTLD. While these bacteria appear to enhance the virulence of this disease, These pathogens that right now are thought to be opportunistic pathogens that colonize the corals after the disease has first become established, and they are not the primary pathogen. This is based on the fact that the type of pathogenic bacteria that we've been identified appear to vary depending on the species of coral, not on whether they have the disease or not, and they are not always found in every coral that shows signs of SCTLD. Through this same effort, researchers have also identified 600 different strains of bacteria within coral tissue that has been taken from healthy corals that appear to produce compounds that kill other bacteria. There's been one strain in particular that's been identified in large cup star coral, or Monastrea cavernosa, that's been found to produce 3 different compounds, which when incubated with diseased corals, has been shown to arrest or slow the disease progression. This compound shows promise as a treatment for diseased Monastrea cavernosa colonies. Basically, if you place a healthy fragment that's been treated with this strain of bacteria adjacent to a fragment that shows signs of disease, it's been shown to arrest the disease progression. While this is a huge advance in terms of developing a treatment, the problem is that this particular strain of bacteria only occurs in colonies of Monastrea cavernosa, and we still need to do a lot more research to further refine this approach before we can effectively use this as a treatment on a reef-wide scale among multiple species. So in conclusion, we have not yet identified one or more pathogens that are the sole cause of SCTLD. We know that the disease is infectious, and it can be transmitted through direct contact and also through water circulation. Different species that are affected by this disease show different responses to the disease, and even within individual species, the rate of tissue loss can change over time. We have found an association between pathogenic bacteria and the disease, and we can treat corals that show signs of disease using antibiotics. We've also identified several strains of bacteria that may produce compounds that can kill the pathogenic bacteria, and with more research, it may be possible to develop a probiotic treatment. So I just want to end my part of the talk by acknowledging all of the numerous partners that have been involved in the disease response. This has been a multi-agency effort involving over 60 partners to date. And you're going to hear a lot more now about the implementation of our coral disease response from Maurizio. Thank you.
All right, thank you so much, Andy. We're now going to hear from Maurizio Martinelli about the Florida management response to the disease.
Thanks very much, Liz, and thanks, Andy. So now that we know a little bit about the disease and the impact that it's had in Florida, I'm going to be sharing about what we've been trying to do about it here in Florida. So I'm going to share kind of 3 main items. The first being, again, reiterating a bit about the partners that we have involved. Then the bulk of the presentation is going to be about the management structure that we have here in Florida, including our response teams. And finally, 1 slide about some of the funding that we've had applied to this disease response. So to start, as Andy has mentioned, we have a ton of people that have come together in Florida to work on this disease. Over 60 partners to date, so as you can tell by this is not everyone who's been involved. But I first do want to again highlight just how many folks we have participating in this response. It is really incredible how many people have come to the table to work collaboratively towards this response. But also, you might notice there are a lot of different kinds of organizations here because there are a lot of different aspects that a disease response requires. So we have 4 lead agencies. That's the Florida Department of Environmental Protection, the Florida Fish and Wildlife Conservation Commission, the National Oceanic and Atmospheric Administration, and the National Park Service. We have additional participation from other government agencies, including EPA, Smithsonian, USGS's National Wildlife Health Center, and more. We also, importantly, have participation from all 5 of the counties in Southeast Florida. And they have really been instrumental in garnering the attention and the funding that this disease response requires and deserves here in Florida. We have almost innumerable academic and research institutions, as well as NGOs, nonprofits, and aquariums, who are all participating in the various aspects of this disease response. So as you can imagine, with this many partners at the table, it can be a little bit difficult to get all the work done. So we recognized as a community that we needed some sort of structure to this response. And again, as a community, decided to organize ourselves according to response teams that focus on specific priority topic areas. So the org chart kind of starts with our leadership team. So our executive coordination team are made up of representatives of those 4 lead agencies. And they kind of give us the high-level guidance that we need for this disease response. Then we have the disease response coordinator position and the new National Coral Reef Management Fellow who work to kind of herd all of the many cats. We really need to make sure that there's information flowing both to and from that leadership body so that they have the information necessary to make their decisions, but also that their decisions are executed by the teams that you're about to see. We also have a technical advisory body, which we call our Disease Advisory Committee, that serves as an open venue for kind of cross-cutting conversations from all of the different partners that you just saw. And then we have our 10 teams. I'm going to walk through each of these teams individually, but wanted to give you an idea sort of what Florida has identified as our priority topic areas. So to start, we have our management team. This is the team that really thinks about higher level issues like over the entire response, where are the resource gaps, what are the funding opportunities and potential allocations, and what materials are we creating, like action plans or communication documents. It is an opportunity for especially our place-based managers to provide their perspective on the locations and also the resources that they have jurisdiction over, and to think through some of the risk management aspects of some of the activities that we're trialing here in Florida. It is also where our county representatives participate most, so they're able to take the information that they gather from this team back to their county commissioners to use as needed. Next up, we have our reconnaissance and intervention team. For the recon side of the house, these are the folks that are going out to identify specifically where the disease is and where it is not. This is essential for identifying the locations for some in-water activities like intervention or coral rescue. Then on the intervention side of the house, this is the team who is going out into the water to conduct the novel coral disease treatments that we've been developing here in Florida. And some of these folks are also trialing and developing the new treatments. We currently have 2 treatments that we're applying. Both are treating those individual lesions that Andy had shown, which is a process that's both time and resource intensive. So as Andy alluded to, in collaboration with the research team, our interveners are thinking through approaches that we can treat whole colonies to hopefully scale up intervention to the site or reef level, or maybe even to larger geographic areas. So we then have our coral rescue team. These are the folks who are leading an effort to collect colonies of susceptible species in advance of that disease margin that was identified by the recon team. The idea is to bring them out of this system and keep them in land-based facilities for future propagation and restoration efforts. One of their main goals here is to preserve the remaining genetic diversity that's in the system. So this team is targeting 50 unique genets, which are genetic individuals per species. This project also includes developing the genetic markers for these species so that we can tell whether or not they're genetic individuals. But since we don't have that information yet, the team is actually collecting 200 colonies of each of the target species in order to hopefully capture that 50 unique genetic individuals. So the first phases of this collection targets 15 priority species. So considering the 200 colonies for each of those, that's 3,000 colonies. And for those who have aquaria, you know that this requires a lot of space. So we are partnering with the Association of Zoos and Aquariums, which is a national network of zoos and aquariums, to help identify facilities that have the experience and expertise and also the space to keep these corals and keep them alive. As you can probably guess, we can't fit all of these corals into our facilities in Florida, so we have been looking outside of the state as well. And just this past month, we've had some of our first rescued corals traveling across state lines. So in addition to the corals that we have here in Florida, we now have colonies that are living happily and healthily in Iowa, New Jersey, and Texas. And there would be no point in collecting all of these corals and keeping them alive for however many years if we don't have a good plan for how to use them in future restoration efforts. Due to the longevity of this disease, especially in those endemic areas, we really need to figure out how to do restoration in the context of this disease. We want to make sure that we are not just putting out corals to die, and also that when we put them out, we're not negatively influencing any of the disease dynamics by, for example, just introducing new material for the disease to flare up onto these reefs. So we have a restoration trials team that is pulling together all of the local restoration practitioners to work together to identify what knowledge gaps we have and figure out how we can answer some of the key questions about restoration in the face of this disease. And they can also start to think about where can we put in effort and resources to building out the restoration infrastructure, because the disease has had such a significant impact here. We're going to need some very significant restoration to follow it up. Most, if not all, of what Andy just presented came from partners within our research and epidemiology team. This team intends to think through kind of holistically what we need to research about coral disease in general and specifically about SCTLD. And then with these needs identified, the team can start to think through how they can best address these research gaps. I also like to point out that this team is incredibly collaborative and collegial. Like, more so than anything that I have seen previously, the researchers on this team have been incredibly open about their projects, constantly updating about what is going well and what isn't, sharing data as soon as it's gathered, all of it. And I really think it was the urgency of this disease really galvanized the community. But seeing their collaboration and cooperation with one another is something that to me is really inspiring and maybe even heartwarming. And I do want to give a quick shout out to our previous speaker, Andy, for leading that team, because it is our largest team and has a lot of strong personalities, and Andy does a really amazing job at herding all of those cats. And in contrast to that team, we have a regulatory team, which is made up of only 4 members. So I like to say it's our most streamlined team, but they aim to ensure that all of the various responsible agencies are comfortable issuing permits for our activities, but also that they're permitting the same activities. They are also working together to ensure that everything we're doing is within regulatory compliance, and where necessary can start to think through how we can streamline any of the permitting processes for our activities. We also hope that this can be a venue where they can think through ways that permitting requirements can benefit the disease response. So, for example, in Southeast Florida, we have some very large coastal construction projects, and we're thinking through, well, maybe some of the permit requirements associated with that can be kind of levied to the benefit of the disease response, maybe, for example, taking corals of opportunity and giving them to researchers or the restoration practitioners. Now, we also have a data management team. As you can imagine, with all of these folks and teams working in their priority areas, we are generating a lot of different kinds of information and data. And so this team works to understand the data needs and the data production of each team and helps those teams to collate and visualize that data. The first priority for these teams is to synthesize and visualize this data for the response partners, but they've also started to consider visualization for other audiences. So, for example, the images that you see on the right show a coral rescue dashboard. This is information on, for example, how many corals have been collected, what species have been collected, where are they housed, and they can even map out where they were collected from. And this can be a very useful tool, so that we all know what's going on, but also, for example, for some of the aquariums that are taking in these corals, they can use this dashboard as part of their exhibit to teach about what we're doing here in Florida. And related to information sharing, we have a communications team. This is our team that works to create the communication materials and messages that our response partners can use. The idea being that with so many different partners, there can be a huge benefit from all of us sharing a common message. Essentially, there's like strength in numbers. So the comms team can help us organize not just those materials like briefing materials, but can gather, for example, all of the speakers who can share about different aspects of this disease response. And they have been and can be a huge help in answering media requests for some of us who have never really dealt with media before. One of the major communications tools that they've created is a publicly facing website on the FKNMS page, which you can see there on the right. This is our so-called disease portal that has general audience-friendly information about the disease and the response, about ways to get involved, images and videos, and even a little area for news updates. So if you can see right there, we can share about webinars that are coming up, like the one that we're all on right now. Related to communications, we realized that there were going to be certain folks or certain sectors in Florida who would be interested in more active participation in the disease response, who wanted to feel, I guess, more ownership over the response. So for us, that's primarily our dive and snorkel community, but we wanted to make sure that these folks had an opportunity to provide more meaningful contributions to the response, which is why it's really citizen engagement, not just communications. So one example is, as you can see on the top right there, there's a little coral with a tag on it. And so our intervention teams, when they go out and treat these corals, they put this little cattle tag on it with a note that says, take a picture of this and upload it to a specific website. And this can be used by your intervention practitioners to help track how their treatments work through time. We can also have these citizen scientists help us with monitoring both where the disease is, but also perhaps post-disease recovery. And finally, we have a Caribbean cooperation team. I apologize to our next speaker, Dana, for some of the spoilers here, but this is no longer just a Florida problem. Unfortunately, other locations in the Caribbean have started to see very similar disease signs, and so we wanted to make sure that there were open lines of communication both to and from Florida. The idea is that while we have been dealing with this the longest and have a lot of information to share, we don't have all the answers. We know that there's plenty to learn from Florida, both from our successes and our failures, but we are not the be-all and end-all of SCTLD response. So we wanted to make sure that any new research or treatments or trials that are either conducted here in Florida or elsewhere are shared back and forth, and not just with us, but with other folks in the region dealing with this disease. Pretty much the more minds and the more hands that we have working on this disease, the better. So those are our 10 teams, and I want to bring us back to the org chart briefly, just to kind of think through how all these teams work together, because I hope you start to notice through all of these different teams that none of them, while they're each their own unit, none of them really work in isolation. So for example, our recon team is essential for our coral rescue team to be able to identify the sites where they can perform their collections. Or for example, our research team, what they learn about disease epidemiology can be crucial for designing the restoration activities in the face of this now endemic disease. Or the summary visualizations that are created by the data management team can be shared with the communications team, who can then work with the other teams to figure out how best to message or disseminate this information to specific audiences. So this is all to say that, you know, none of these groups work in isolation, and it's really important to have that collaboration across all these different areas. And I'm going to finish up now just quickly talking about funding. And I want to note from the outset, this is not a comprehensive list of all of the funding that has been put towards this response. It's just some of the major pots that jump out. So the largest appropriation has come from the Florida State Legislature. If you'll recall from the beginning of the presentation, when I mentioned the importance of the counties, it was really those county reps that were instrumental in raising the alarm at the state level that led to this kind of support. So since the mid-2017, there has been increasing appropriation from the state that's kind of funneled through DEP that has gone to many, many different projects. We also have support from state agencies. So FWC through their state wildlife grants and DEP through some internal funds shuffling have put money towards research and other projects. On the federal side of the house, EPA has supported the response through two mechanisms. First, their wetland program development grants, and then just their research grants. And NOAA, the Office of Coastal Management, and their sub-office, the Coral Reef Conservation Program, have allocated funding primarily for dedicated positions related to response. And NOAA Protected Resources has helped fund aspects of the coral rescue. And finally, NIFWIF has been the primary source of funds for the coral rescue, and NSF has supported, as you might assume, some of the research, and this was through their RAPID grant. Now, I want to note a big area that's missing here is staff time. We sometimes joke that most people involved in this response still have their day jobs, which is to say that we have very few folks who are funded exclusively to work on this disease, and everyone has had to sort of reprioritize and reorganize, which is no easy feat. So I really want to make sure that that is recognized when we think about the resources that are put towards this disease response. And with that, I'd like to say thank you, and I guess I can throw it over to Dana to talk about the Caribbean.
Great, thanks so much, Maurizio. Oh, go ahead, Dana.
Thank you, Maurizio, very much for that. I'm going to talk about a collaborative effort between those involved in the Florida disease response, as well as key partners who operate at a regional level in the Caribbean to provide support, information for the prevention, detection, and response to stony coral tissue loss disease. And I'll run through some of the resources and materials that are available to you. So first, to add some logos to the salad of organizations that Maurizio presented and Andy presented to us, where we are working with some key regional partners who work closely with coral reef resource managers, scientists, and practitioners in the region. And they have been able to get quickly involved and provide some really key support for both the detection and response to SCTLD in the Caribbean region. As Maurizio mentioned, we have one of the 10 response teams in Florida is the Caribbean Cooperation Team. Here we all are. I co-lead the team with Judy Lang at AGRA and Jennifer Koss, the director of NOAA's Coral Program. We have representatives from those regional level organizations and some of the key players in the Florida response. And we're really trying to serve as a conduit of information back and forth from the region, both to help track and collect information on disease observations, vet them with our scientists to see if they match the stony coral tissue loss disease case definition. Also to track the intervention and treatment approaches being tried so we can be sure to share information on what's working and what's not working. And then, as Maurizio said, to share lessons learned from the Florida experience, to share those successes and failures at our attempts to curb this new disease in the region, and to share some of the materials that we think can be of use to our colleagues in the region. So the folks at AGRA have developed an online database that I will get to in a couple minutes here, but they generated this map and are able to track and make publicly available the locations where stony coral tissue loss disease is now observed. So after the disease spread along the Florida reef tract, it started showing up in the Caribbean region. We first saw it in Jamaica, then along the Caribbean coast of Mexico in the state of Quintana Roo, followed by St. Martin, the southwest coast of St. Thomas, and then most recently in the Dominican Republic. We have received reports from other locations that after reviewing photos and data submitted, we were able to call false alarms from places like St. Croix and St. Eustatius, the Cayman Islands, and in Culebra, Puerto Rico. And we have some anecdotal reports coming in from places like Haiti, the British Virgin Islands, Turks and Caicos, Curacao, Aruba, and most recently Isla Alacranes, a protected area in Mexico, in the Gulf of Mexico. And we are hoping to receive some photos and more information, either to members of the Caribbean Cooperation Team or through AGRA to verify whether or not what they are seeing looks like SCTLD. But I can't emphasize enough how much we need everybody's eyes out there on the reef sharing information about what they're seeing when it comes to disease on our coral reefs in the Caribbean region. It's not advancing. Okay. My presentation seems to be stuck. Oh, there we go. Sorry about that, everybody. One of the organizations that is heavily involved in the region, specifically in the Mesoamerican Barrier Reef area, is the Healthy Reefs Initiative. They've documented the rapid spread of this disease along the Mexican Caribbean coast. It was first reported in Puerto Morelos, which is just south of Cancun, last July. And by December, it had been observed from Cancun in the north all the way down to Ishvilak at the very bottom of the coast of Quintana Roo, and Ishvilak borders Belize. So it had spread over 400 kilometers in just 6 months' time, and it's now estimated that 30% of the colonies from those affected species have been lost. We'd like to share some resources that have been developed to help with prevention of the spread of stony coral tissue loss disease. Knowing that it is waterborne, one of the first things we wanted to do was to make sure that the disease is not being inadvertently carried on dive gear by our science and recreational divers. This is a decontamination protocol that was developed and is available at the Florida Keys National Marine Sanctuary Disease Portal That site is on the bottom here, the link, and we will provide it at the end of the talk here today. But we provide some recipes for decontaminating dive gear. For non-sensitive dive equipment, a 10% bleach solution followed by a freshwater rinse and a good air dry. For more sensitive dive gear, using ammonia-based solution, again followed by that freshwater rinse and making sure it's dry before getting back in the water if possible. And then for sensitive equipment, an antibacterial soap-based solution with a freshwater rinse and the air dry. And again, always trying to, if possible, make sure you're not diving from disease sites to non-disease sites before disinfecting your gear. Also understanding that this is a waterborne pathogen, we must consider ballast water as a potential vector of the pathogen. Rob Brumbaugh here, who on our Caribbean cooperation team, is our point person on ballast, and he put together some information to share today to help everyone understand how ballast is regulated through the UN International Maritime Organization. New regulations entered into force into 2017, and those are enforceable. In the US, we have recently developed new legislation and have 5 years to implement those in the form of regulations. Many have probably heard that the Dutch Antilles have taken a precautionary measure and are prohibiting the release of ballast from ships that are coming from countries where SCTLD has been identified. And if you are able to consider taking similar precautions, you might want to consider that different types of ships carry different quantities of ballast. So focusing on those larger bulk carriers, they would carry and release the most ballast, considerably more ballast than a cruise ship in the passenger category at the bottom. We'd also like to share some resources that are being used and available to help with the detection of stony coral tissue loss disease. As Andy highlighted earlier, there are some species that demonstrate, of the susceptible species, that demonstrate signs of disease earliest. And so looking at the maze corals, the pillar corals, the elliptical star corals, and the smooth flower corals, and then the brain corals are the first species to show those signs. And this one-pager was developed by MPA Connect. That's a partnership between the Gulf and Caribbean Fisheries Institute and NOAA's Coral Reef Conservation Program. And this is a handy one-pager that can be printed out and shared. And as you can see, it provides good information on those species that we first see the disease in, explains the difference between this disease event and other coral reef diseases that we've seen in the past in terms of the rapid spread and that high prevalence and mortality rate. As a part of the Florida response, Andy created these the stony coral tissue loss disease ID cards. These can be downloaded, printed, laminated, and brought into the field. There's pictures of all of the affected species. You can see what a healthy colony should look like, and you can see different ways that stony coral tissue loss disease can present itself in that species. One of the things that's useful about this ID card set as well, and it's 3 double-sided cards, is the final card shows other conditions affecting corals that may get confused for stony coral tissue loss disease, like bleaching, predation, growth anomalies. And so you may want to look at those and consider whether or not what you're observing is one of these conditions. Dr. Karen Neely, who heads up our reconnaissance and intervention team here in Florida, developed a slideshow to help with the identification of stony coral tissue loss disease. This is available on the AGRA website as well. And this is meant to be a slideshow. You sit in front of your computer and look at the pictures of how stony coral tissue loss disease appears in the affected species. And then we have a monitoring methodology that can be shared if you would like to conduct a survey to look for stony coral tissue loss disease. There's a methodology and a downloadable data sheet that you can access. AGRA has it up on their disease webpage to conduct a roving diver survey for stony coral tissue loss disease. In terms of where to look, you want to focus on those areas with a high abundance of susceptible species. If you collect AGRA data and submit your data to AGRA, they can complete an analysis for you and help you identify where these locations might be. You also want to look in areas of high shipping activity, the vicinity of ports, areas downstream, areas where ships are anchoring or where lots of ships are passing through on their way to and from port. Here's an example of the analysis that AGRA is able to conduct to help pinpoint areas where you might want to do reconnaissance monitoring. They did this analysis for Belize to highlight for them where their highest populations of the Orbicella species are located. And they can also do a similar analysis to find the larger colonies. Now, once you've observed coral reef disease and stony coral tissue loss disease, or what you think suspiciously looks like stony coral tissue loss disease on your reefs, where do you report that? You can definitely send information, photos, and a description of what you've seen to any of us on the Caribbean Cooperation Team, but we also are directing people towards this great new database that AGRA has developed. There are 3 ways to submit that data, whether you were just out doing other monitoring work or whether recreational divers were out diving and casually observed disease and took some photos. There's a place to submit those. You can, as a science diver or a member of the scientific community, you can submit information by species name. As a recreational diver, you can submit information using the common coral names. And then if you actually were able to plan and execute a roving diver survey, you can submit your data from that survey on this website as well. And if you've already collected data and you've used a different methodology, you can send an Excel file to the AGRA folks, and they will make sure that your observations are documented on this database. So we're all looking forward to using this as a regional tool to help us track the disease. Now, there are some resources available for response to stony coral tissue loss disease. We have a disease intervention action plan developed by Karen Neely in Florida. This includes the methodologies for the intervention approaches that have been used along the Florida Reef Tract, as well as the recipes and supplies that are needed to create the treatments. As Maurizio said, the research community has been very forthcoming with their data, and the Florida Department of Environmental Protection is tracking all of the research projects associated with this disease event on their website, and you can access those studies and the outcomes of those studies there. And there's been lots of training and exchange of knowledge going on related to this tissue loss disease. Back in December, MPA Connect hosted a training event for marine protected area managers in Mexico and brought them to the Florida Keys. Karen Neely provided training in those intervention and monitoring techniques, and they were able to go out and see affected corals on our reefs here. Healthy Reefs has been monitoring the spread of the disease, has been applying treatments to diseased corals in Mexico, has been conducting outreach workshops in Mexico, and has helped develop some outreach products. They also recently conducted a training in Belize on stony coral tissue loss disease. Again, the disease has not been observed there as of yet, but being so close in Ishkalak, they are getting prepared for it. And then just last week, we had a great group here from the U.S. Virgin Islands and held a learning exchange on the disease. And we're able to bring them out into the field and show them the intervention and monitoring techniques we are using here in Florida as they prepare to intervene in St. Thomas and prepare for that disease to show up in St. John and St. Croix. And so I think Liz is also going to continue showing these websites up on the screen, but here are the links to the key websites that are housing all of these information and products. Thank you, everyone.
Great, thanks so much to Andy, Maurizio, and Dana. I will go ahead and share my screen again. 1 second. And we're going to go ahead now, and we have some time for question and answer. And we have a couple of people. Let me just make myself presenter again. Okay, so as I was saying, we have a couple of minutes left for questions, and we've gotten a number of questions in the question box already, which is great. You can also ask questions by raising your hand. There's a little hand icon next to your name, and I will go ahead and unmute you to ask your question. So the first one that we got says, Maurizio mentioned a lack of space for coral rescue and restoration. I think that means lack of space or space needed for putting corals into aquarium. And this question asks, what is required to be a part of this effort to provide aquarium space? This person has some experience with an aquarium in Chicago and would be happy to be a contact person with that aquarium and is interested in being a part of this process. So if you have any information, Maurizio, on becoming part of that larger effort, I think that would be great.
Sure, great. Thanks for the question. We have primarily been working through AZA, that Association of Zoos and Aquariums, because they are this national network and we're kind of tied into a lot more facilities than we were. There are specific requirements related to essentially biosecurity, because we want to make sure that we're not putting any potentially diseased corals into an open system that can then impact any of the other corals that are kept there. We also can't put them into tanks with Pacific corals, for example, because then there are concerns about if we then outplant them, if there's anything that's being transferred out. So our coral rescue team has a number of different requirements for those facilities. But if you reach out to me, I can definitely put you in touch with them. We'll get you the information that's needed to see if your contacts in Chicago kind of can get looped in as well. So that's great. Thanks very much.
Great, thanks so much, Maurizio. Our next question asks, I have heard reports coming from Bonaire as well. Who would be the contact person, or how would I submit pictures and information?
So the best place to send your information is that AGRA database. And so Liz, are you able to show those web links again on the screen so that everyone can be sure to be able to get those? Or
Sure, there we go.
Great, perfect, yes. The AGRA database is the best place to submit your information, but you can email any of us on this call here and we will get that information to both to AGRA as well as review any photos you're able to send and help you discern whether or not it looks like SCTLD.
Great, thanks so much. We have a question from someone that has their hand raised. Rebecca Gibble, I'm going to go ahead and unmute you, and you can go ahead. Go ahead, Rebecca.
Hi, I was just wondering whether it's possible to purchase the disease identification cards that Andy Bruckner prepared. They look really useful.
Those are free. Those are available. They're free as a digital download, and so you can access those on any of these websites up here. They all have them posted, or definitely the Sanctuary one and the AGRA database one do. We have not produced them for dissemination as field cards, but might be able to do that in the future if you are unable to print them out and laminate them yourself.
Yeah, probably laminate myself. Thank you very much.
Sure.
Okay, and we have another question in the question box. Does this disease seem to have a depth limit, or is it too soon to tell how mesophotic reefs may or may not be affected?
So, I mean, I'll try to answer that. Within Florida, we've only been looking to about 80-foot reefs that are about 80 feet deep, and we do see it. Those susceptible species occur to all those depths. There's a large concern that's been raised within the Virgin Islands in particular because they have a very large mesophotic reef community that's dominated by these star corals or Orbicella, and that's one of the healthiest areas left there. And so we don't know yet if it's going to spread to those particular species. We hope not.
Great, thanks so much, Andy. We have another question from someone, Melanie McField. I'll go ahead and unmute you. Go ahead, Melanie.
Okay, hi there. I guess this is probably to everyone, or maybe to be deflected towards the communications team, but I think it's so important how we communicate this out to the world, and the seriousness of this disease. I can't wrap my head around calling it SCTLD. I mean, I think it really is an important thing, what it is called when we communicate this out. I see a little pack of Skittles, floating up into the sky. I want to like put in a request that you guys go back to the drawing board and think about what to do. What we've kind of been calling it for stony coral tissue loss disease, STD. And somebody used that they called it like the HIV of the reef or something. And I think that would be a more effective way to like convey this. So anyway, that's just been bothering me the whole time. And I just wanted to put it out there for you guys to think about.
Thanks. We have a communications team call in a couple days, so I will raise that with the group.
Thanks, thanks, Maurizio. Okay, we have another question from the question box. Is the probiotic a problem for fish or any other organism on the reef?
So we don't know yet at this point in time. So anytime that you do anything in the field, it requires really a lot of laboratory work, a lot of testing, and then you need to try something out sort of at a pilot scale. We know that probiotics have been used in aquaculture facilities, so a closed system. One of our biggest challenges with coming up with a treatment that's very effective in the ocean is because there are so many reefs and the ocean is wide open. We have to be really, really careful what we put into the water. So it's quite a long ways down the road initially, but what I envision is it's not going to be released just in the water. It's the same way as our antibiotic trials. We're using a very, very low amount, and it's put in direct contact with the diseased lesion. So it's not just, freely added to the water. And I think that's a really critical thing that we have to be careful for because we don't want to have any impacts on any of the other organisms.
Great, thanks, Andy. We have another question from someone with their hand raised. Heather Stewart, I'm going to go ahead and unmute you. Go ahead, Heather, and ask your question.
Okay, well, thank you guys, all of you, for your presentations. I'm really concerned about this because I do my work in multiple regions, and I'm concerned about bringing stuff, especially since I'm starting in Florida and then going to the US Virgin Islands and then down to Panama. And so I don't want to bring anything with me. So I was going to use the disinfectant regime that you had suggested, but I know there's also like different tablets that you can add for cleaning gear as well. What are your feelings about like certain precautions in order not to carry this disease with us as researchers?
You want to take that one, Andy?
Sure. There's, so we're running into sort of the same issues here. First, who can treat their gear, how to do it, especially for a researcher, because in a lot of cases when you go diving, you may do bounce dives and hit multiple regions, and there's been concerns that it's not practical after every dive to treat your gear. One of our biggest concerns is not transmitting it from one region to the other, and so that's where the decontamination protocol is really, really important. The protocol that we developed was basically looking at all other protocols around the world that have been used to prevent the spread of pathogens, both for corals as well as other diseases that occur in aquatic environments. There's a lot of other compounds that have been identified, and it's something that we're interested in doing more research on to see which ones are effective. There's certain gear types also that are more likely to serve as a vector for some of these pathogenic bacteria. In particular, your wetsuit material. The bacteria tend to stick to that really well, as well as the inside of your BC, because that stays moist for a long time. And if you really focus on those components, that's probably the most critical thing you can do, as well as any tools that you use. And then the other thing that we really try to do is when we are diving in an area, we try to dive the site that is presumed to be unaffected by the disease first and hitting the disease site last, so that we don't, take a chance of, diving somewhere, working with the disease, and then moving into an unaffected area and transmitting it to that area.
Yeah, and that was my concern, is because I don't have the control of doing Panama first because I'm already in Florida. So we were going to do bleach for everybody's gear, and then we're going to have like a really thorough decontamination in between regions, and we're going to use, I think it's Viricon S. Do you know anything about that product or how well it works, or is it just as good?
Yeah, so we've actually recommended that product for more sensitive gear. So the problem is if you use bleach on your regulator, anything with silicon, if the concentration is too strong, you can actually start to break down the silicon material. So that's why we have this sort of three-step process.
Okay.
The compound that you're talking about is from our tests, it seems to be very effective on wetsuit material. It's just really, really important that after you soak it, that you really thoroughly wash your gear with fresh water, and then you let it completely dry. So that adds a second layer of protection.
Okay. Thank you.
Great, thanks, Andy. And then we have another caller, Jessica Burkle. Would you like to go ahead and ask your question now? You're unmuted.
Yes, my question was looking at the slide that had the photo that had the person using the grinder underwater, and also when you're about to apply the treatment. I'm just concerned about tissue being released into the water. So is the recommendation that I just let it die, or do I try and treat it and thereby release some tissue into the water? That's my concern.
Hey, this is Maurizio. I can maybe start to field that question. For that protocol, that where you see the grinder being used is creating a fire break within what we think is healthy tissue. So we don't think that we're adding disease tissue up into the water column, because we're cutting into where we don't think the pathogen has made it yet. There are definitely concerns if we were going to do that right on where the disease margin is, that we're adding, more pathogenic material into the surrounding environment. But the hope is that doing it within that healthy tissue, we're not doing that. It is definitely a concern, and one of the reasons that we want to move away from these sorts of lesion level treatments, so that we can do all that we can to treat these corals more holistically.
Yeah, and I can just add one more thing to that. One of the actual recommended steps if you are going to create this fire break, is that you actually have an aspirator underwater with you. Years ago, within the Florida Keys, we treated a lot of corals with black band disease, and we did the same sort of thing. You were basically removing the black band material and some of the tissue and skeletal material, and we had a very strong aspirator, so none of that went up into the water column, because that could be a huge concern, that you could actually make the disease worse. If you tend to be grinding and you get into an area where there is the pathogenic organism, then you could be spreading it that way. So really, I would recommend, if you're going to do that, that you use some sort of aspirator to suck all that material up as you're grinding.
Great, thank you guys. There's been a couple of questions in the question box that are related to whether we know about environmental correlates to this disease. Is it related maybe to human impacts or any other relationships that you're seeing
So I can at least start, and then Maurizio and Dana can fill in. But when this disease first occurred, in one of the publications that's come out, they tried to link it to the 2014 bleaching event. We have seen in other areas in the Caribbean and other parts of the world that when corals bleach, they may potentially survive and start recovering from that bleaching, but their resistance is down and they're more likely to succumb to some sort of bacterial infection or disease caused by something else. And we saw that after the 2005 and 2010 bleaching events where there were these large outbreaks of white plague. We don't think that that was the sole cause, because if that was the case, we should have seen the disease all the way throughout the Florida Keys, whereas when it first appeared, it was in one very distinct, concentrated area and not everywhere that bleached. So the bleaching could have been one thing that just weakened the corals. And we think that a lot of other environmental variables may do the same thing. Stressful conditions besides high temperature, sedimentation, nutrient loading, all of these things could weaken those corals and cause them to become more susceptible to the diseases. What we don't know, if it is a pathogen, if that pathogen came from somewhere external, or whether that pathogen was already present on these corals and these corals basically just became compromised, allowing those particular pathogenic bacteria to take off and take over the bacterial community and cause the disease. We don't quite understand that yet. We don't know where the pathogen could have come from. But one of the things that we're really looking at is addressing everything from water quality issues, any sort of local human impacts are likely to improve the condition of those of the reef environment in general and make the corals more resistant and resilient to these diseases.
Thanks, Andy. I think you hit the nail on the head. One thing that I want to add is that a few years ago, we had an effort that was trying to collect all of the essentially environmental data sets that we had for the area around the epicenter of the disease, so around Miami, to see if we could start to pick apart what were some of the environmental conditions that led to perhaps the outbreak of this disease. Unfortunately, there was so much going on. As Andy had mentioned, there was this thermal stress event, there was sedimentation from a large-scale dredging project. There's evidence that we had one of the sewage outflow pipes that goes out past, it's supposed to go out past the reef, actually had a crack in it, so you have this introduction of treated sewage right near the site. So I like to think of it as there's kind of this soup of conditions that all came together. Which is a long way of saying we don't know, we've tried to look into it, and I think, as Andy said, one of the best things that we can take from this is to do all that we can to improve those larger scale environmental conditions, to make sure that the corals have more of an opportunity to recover from this disease outbreak, but also so that nothing like this can happen again to what remains on our reefs.
Great, thank you guys so much for answering all those questions. We've gone over a little bit and there's still a lot of questions that people have. And so here's a list of resources that Dana shared earlier. The Reef Resilience team is actively working with these presenters and many other people to put together a page of resources that will be available on our website. So stay tuned for more on that. And then also we are recording this webinar. There was a couple of questions about getting the slides from this webinar. This webinar will be recorded and posted and always available on our website as well. If you go to reefresilience.org/webinars, you'll find them there. We also send it out in our newsletter directly after this. And for all of those additional questions that we still have, we'd like to move the conversation over to the network forum. This is an online open access community for coral reef managers and practitioners. We have people from all around the world who are part of this community. So we will post all of the questions there, and our presenters from this webinar have agreed to spend a little time on there and answer those questions and do some follow-up with you there. So please take advantage of that opportunity. There's some instructions here on the left of the slide where if you are not part of the network forum yet, you can go ahead and join. Just go to reefresilience.org and then you can click on the network forum. There's also a link there in the top right corner of the slide you can just go ahead and apply or submit a registration form. Your membership will be approved, and then once you're in there, the first topic you'll see in the forum is dedicated for this webinar. So we'll post the resources there, the list to all those links. We will post a link to the recording of this webinar, and we will post all of your questions that we didn't have time to get answered. So I want to thank you all so much again for attending this webinar, for all of your really great questions, and thank you again so much to our really excellent presenters, Andy, Dana, and Maurizio, for sharing so much information. And we look forward to seeing everyone in some additional Reef Resilience Network webinars in the future. Thank you very much.